here is the monomer of cystatin C, and that is the most basic form of the protein, which is actually functional. What we see here is a lot of the actual secondary structures which we previously discussed, such as our 5 purple strands, each corresponding to beta strands 1, 2, 3, 4, and 5, together forming a larger beta sheet. Within the center of our protein, we can see a large corkscrew structure. This is our alpha helix. Alpha helix 1 has 5 turns, and it occupies the center of our protein next to the faces of the beta sheet. We also have a secondary alpha helix off to the side, and this is much smaller and it is more offset compared to the rest of our protein. What we also observe here are the N-terminus and the C-terminus of the protein, meaning that if we were looking at the primary structure, all 120 residues stretched out, this would be one side and this would be another side of the singular chain. Some characteristic features of these secondary structures which contribute towards its three-dimensional folding is the fact that at the core of the protein, this environment is very hydrophobic, meaning that it's non-polar. The reason why this is that this face, the inner face of the beta sheet which faces the alpha helix, and the alpha helix face which faces the beta sheet, contain amino acids which are very non-polar. And as like attracts to like, this forms interactions which attract the two secondary structures together, holding the core of the protein together. This is very important to the three-dimensional folding of the protein and its function. Furthermore, what we observe is that the N-terminus with this loop between beta 2 and beta 3 and the loop between beta 4 and beta 5 actually form a hydrophobic wedge. So these three structures here together, where these two loops between 2 and 3 and 4 and 5 are referred to as beta hairpin loops, will form a binding site which is very hydrophobic, and it's very complementary to the hydrophobic binding site of cysteine proteases. That means that the combination of these three structures, N-terminus and then loop 1 and loop 2, will be able to bind to the cysteine protease, occupy that active site, and prevent other proteins from binding to the protease and being degraded. And as a result, this three-dimensional structure of cystatin C is what contributes to its inhibitory functionality.